This webinar is an introduction to the second community challenge that we have on Mosaic, the Clinical Strain Detection Challenge, also referred to as Strain number 2. We're excited to have with us today Dirk Gevers from the Janssen Human Microbiome Institute to give an introduction to this challenge and the other challenges we have going on. The second speaker is Keng, who will dive in the details of the strains detection challenge that we have on Mosaic, and then we will do a live demo with Sam Westrike from DNA Nexus. So my name is Mihaly Sariotomas. I'm the microbiome lead for DNA Nexus, and I also have with me Caroline, who is going to help administering this webinar. So before I pass control to Dirk, I would like to mention that we have two challenges going on right now, strains number 1 and strains number 2. There is still time to participate in strains number 1, so please go ahead and visit the platform to participate. And we also have a new challenge coming up in a few weeks, the standard challenge, which we'll talk about in the end of this webinar. I would also like to remind you that if you have any questions, we'll be pausing between every speaker in order to be able to answer them. So feel free to use the Q&A feature of the Zoom webinar application you have in order to ask these questions. So with that, I will pass control to Dirk. Dirk, can you start?
Oh, thanks, Michalis, and thanks everyone for joining today's webinar. I'm very excited to see people really dialing in and having a great interest in what we're doing. So let me set up a little bit. As the global head of the Janssen Human Microbiome Institute, I've been really exploring the microbiome's potential for translating it into health solutions. And that could go very broadly. But in doing so, I really had the privilege to work with numerous different entrepreneurs that are actively looking at this in many different directions, from bugs to drugs, phages, whatnot, many different approaches. About 150 companies in the US and Europe alone are pursuing this today. I also had the privilege to interact with a lot of scientists that are really pushing the new learnings forward here, and not only discovering the science, but also developing new technologies and methods for looking at the microbiome. And so I've really learned that it's going to take a village to really make the best out of the microbiome's potential. There's great progress, but there's still a lot to be done. And that village is best to be composed out of multiple different players. And so this first slide here shows a kind of a vision for what we have behind the Mosaic platform. We really like to build a community that would bring stronger interactions and partnerships between academia, industry, and even our regulatory agencies. And I believe that we all have partially overlapping interests, that if we were to bring them together, we can make great solutions based on the microbiome that will impact health. I want to stress that we as Janssen do not own any part of Mosaic. It's really intended to be an open and inclusive community that comes together around open data sets, open source tools, and so forth. You know, a couple of things that I've learned from interacting in the past few years in this area is that besides we need a village to come together, there's also computational analytics is going to be really key to unlock the potential of the microbiome. And it's my observation that the current rate of the innovation at which this is happening is perhaps not in par with how quickly we're trying to produce novel products. And I think those products are requiring certain tools and certain types of analysis that we don't really do often quite well today. And advancements, I believe, are needed in precision of those tools, also at the resolution of those tools, in how well they're scalable and usable especially within this industry type of setting. So to make it a little bit more specific, I think one key example here is on strain detection. I think on the academic front, we're all starting to really appreciate that the unit of action in the microbiome world is really a strain, not so much a species. And we're now getting at the point where data generation is really enabling us to look at that finer resolution. But we feel that if we look at the existing tools out there, and kind of a great manuscript referred to here on the right, borrowing a figure from that, if you look at existing tools and how well they perform in terms of species level performance, many are doing quite well, but as soon as you try to use them for strain level, it's performing less well. And at the point where we are concerned, we really want to be able to understand when we plan to add microbial-based products to patients, we want to be able to understand how they behave in those subjects. And we believe that you need to be able to have quite precise as well as strain level resolution methods for that. We launched a little while ago a first challenge, which is referred to as the Strains 1, last December. We are now launching a follow-up on that. In fact, together with the FDA's CIF-SAM pathogen detection challenge, which I believe are very overlapping goals and perhaps even overlapping analytical solutions. I think time will tell, and I very much encourage those interested in this particular clinical strain detection to also take a look at pathogen detection challenge. So just a little bit more about the difference between the 1 and 2. First of all, we believe that one challenge will not be sufficient to really easily resolve the strains issue. And so we really like to build some momentum towards really clarifying how challenging it precisely is, but also then stimulating some progress. And so what we thought was a first good start was to really help strain level profiling, where we don't quite know exactly what is in a sample. We were trying to leverage reference databases, for example, that are existing. So we don't have a preconceived notion of exactly which strains to expect, but we want to get a good sense of what's in there at the highest resolution possible. So that was Strains 1, as mentioned, still ongoing. Strains 2 is a little bit more specific. Now we want to be able to detect clinical strains or even track them. The difference here is that we actually know which strains are in there and which strains we do want to be having a readout. We also want to be sensitive to closely related organisms to these and have a sense on whether they are present and how they, in what ratio compared to the known strains. But you can see that this is very much driven by that initial notion where we have a product with known strains. We're not going to be administering products to patients if we do not have the genome sequence. So we'll start from having that knowledge. And so we thought that perhaps while this is more targeted, this should also a little bit more easier to achieve than the initial strains profiling, but might need some more tool development in the sense that the strain profiling tools out there right now typically work with the reference libraries that exist. Here, I want to make sure that we use the fact that we have those genomes available. More details on that to come in the rest of the webinar. I'll wrap up with this slide here and say that we're now towards the end of February 2018 launching Strains 2. There's more to come. We have a couple of more pieces in the works as Mikhail has already referred to. But I also want to say that we're really excited about the collaboration with DNA Nexus on this, as particularly in all the wealth of expertise that they bring in cloud computing and organizing these types of challenges. And so you see at the bottom of this figure all the pieces that they are currently working on that will be added on top of this platform, and will hope to bring more benefits to the community with time. Last thing I'll say that Mosaic is free for anyone to launch their own challenges. We are taking a stake here with a number for this year, but you should know that we are going to be bringing this capability also to others to really open it up for maximizing what we have been building here. So with that, I'm happy to take any questions if they are rising up right now, or otherwise pass it on to the next one.
Thank you, Dirk. So we'll give a couple of minutes to participants to ask any questions. Again, you can use the Q&A feature of the Zoom webinar application to ask any questions for Dirk. So we'll pause for a couple of minutes right now. Okay, we have one question. Dirk, who has ownership of the IP developed on Mosaic?
It is the developer itself that owns the IP, and DNAnexus is fabulous in if you were to be sharing tools that are developed with this community, is that you actually can use whatever license that you prefer around this tool. And so I think number one, we want to be able to objectively compare how different tools are performing, and you participate with your full name or anonymously. If you were to choose to share tools with the community and think this is a great platform to do so, to make it widely and easily accessible, we would definitely encourage that, and we can wrap that up with the license that the developer prefers.
Great, thank you. Another question, what does JHMI plan to do with the winners of the challenges?
We're absolutely seeking to recognize that in the best possible way, and I'm looking towards the team to see what we today have officially announced as what the prize will be, but we want to make sure that you all know that there are plans to basically elevating the stake a little bit where we can over the next couple of weeks. So I don't know, Michalis, Keng, do you know what the current public statement is on the winners?
So we are this is Keng. Yeah, we are preparing to see if we can put together some kind of prize for it, as well as working with some of the major conference organizers to have sort of recognition or talks by the winners themselves. And definitely we are also pursuing the publication route in terms of preparing publication to come out from the analysis of these challenges.
Right, so I will add to that the publication will also extend to the participants, not just the winners, right? So anybody who participates, if there is a paper coming out of that, if they submitted the results or analysis in the challenge, then we'll reach out to them and figure out how to include them in the publication. Okay, I think that's all for Dirk. We can move to the next speaker, which is Keng, Senior Computational Scientist for JHMI, and he will dive into more details about this second challenge, the clinical strain detection challenge. Keng?
Right. Thanks, Michalis. Hi, I'm Keng. I'm the Senior Computational Scientist leading the Mosaic effort from the JHMI team, and I'll be talking about the design, the evaluation metrics, and the submission format for the challenge. So this is an overview of the challenge. So first, participants will have to create a free account on Mosaic, which would then allow them to join the challenge. And joining the challenge would automatically create workspaces which participants can access the data set and also the other resources needed to participate in the challenge. Later, Sam will give a live demo of the entire process. And we have structured the challenge as a data science challenge by dividing the data sets into the training data set as well as a challenge data set. The training data set is really meant to help participants refine their methods. So we will be providing the truth for this data set when we launch the challenge. And participants can then submit their results for this data set and have immediate feedback on how they perform by submitting their results through the evaluation app on the testing ground. Whereas for the challenge data set, we will only provide the data sets themselves during the course of the challenge. And this, besides minimizing the risk for methods being overtrained, we are also providing a more unbiased way to evaluate the methods. And in terms of how can participants take part in the challenge, there are two ways. The first option is for the participants to choose how to download the data set and then work on them remotely and then submit the results through the evaluation app on the Mosaic platform. The alternate way is for the participants to deploy their methods as Mosaic ads on the platform, and this enables them to leverage on the free computing resources that we have made available for the challenge. So Sam will be going through the demo in terms of how this works out. And just to stress that the final results of the challenge will be based solely on the performance of the challenge data set. And as Dirk has mentioned, participants in the challenge can either publish the results with their real identity, or they can choose to submit it anonymously. The challenge data sets the data sets for this entire challenge was developed in collaboration with Jeremiah Faith from the Eaton School of Medicine at Mount Sinai. So first, we have isolated novel bacterial strains from human fecal samples and sequenced them and then assembled them to provide the isolate genomes. The fecal samples themselves were also sequenced deeply. And hence, the goal is really to be able to match each organism to the source microbiome or metagenomic that it came from. So each of these strains will only map to one sample. And at the same time, we have also provided or added negative control strains in this data set. And these are strains that should have not been isolated from any of the samples. So they should not have a hit at all to any of the samples provided. Next, I will talk more about the data sets that we'll be providing. So in terms of the metagenomic samples, we'll be providing 4 metagenomes each for the training and for the challenge data set. And these are raw metagenomic FASTQ samples that have been sequenced using the Illumina HiSeq 4000 machine. And they're within the 4 metagenomes for the training data set. 2 of these are newly sequenced samples, and there will also be 2 publicly available decoy samples. So the purpose of the decoy samples are to provide samples as negative controls whereby no strains are provided in the genome data set that will be mapping to them. And for the challenge data set, in terms of the metagenomes, we will be providing 4 metagenomes, but we will not be disclosing the number of decoy samples within this data set. And at the same time, we will provide genome data sets comprising of 40 genomes each. And these include strains that are true positive strains that have been isolated from one of the samples, meaning that they are present in one of the samples, or they can be negative control strains that are not present in any of the samples. And our collaborator Jeremiah Faith has taken very detailed care to make sure that the risk of contamination within the samples are minimized. And at the same time, we will be providing the truth sample for the training data set, which will be made available once the participant creates an account and participates in the challenge. And for the challenge data set, the truth will only be made available after the challenge has been completed. So in terms of the submission format for this clinical strain detection challenge, we'll be taking a tab delimited file that can be of 2 different options. So the first option refers to those methods that only predict the presence absence of a particular strain to the sample itself. So if you look at the leftmost column, column 1, it refers to the column ID as given in the reference genomes files provided for the challenge, whereas the subsequent columns 2 to 5, there can be two different formats as described. So in the first format, it will be a binary number, indicating whether the particular strain is present in the particular sample, or 0 for absent, meaning that it was a negative control strain throughout the 4 samples. And for the second format, we will be taking in the presence absence together with the probability that the method is able to predict in terms of whether a particular strain is from a particular sample. And this allows the calculation of the precision recall curve for the submission. Hence, only submissions with the probability would be able to generate the precision recall curve. Next, I would like to talk about the evaluation metrics. So we'll be evaluating the submissions using the following metrics based on the true positive, the false positive, true negative, and false negative numbers from the submissions. And these will be used to calculate the accuracy, the precision, recall, F1 score, as well as the misclassification rate. In addition, we will also calculate the adjusted RAND index, which is the similarity index between two clusterings, considering all pairs of samples and counting pairs that are assigned in the same or different clusters between the submission and the truth. So this adjusted RAND index has been adjusted for chance, and we will also be using the adjusted RAND index to showcase the leading submission in the leaderboard itself. And the challenge is open as of last Monday, February 12th, and we will be closing the challenge on April 19th. And we will be taking the time then between to evaluate the submissions, and we hope to be able to announce the results at the 2018 ASM Microbe Conference. And with that, I would pass the presentation to Sam, who will do a live demonstration on how the workflow is like.
Actually, thank you, Keng. Before we go to Sam and the live demo, I would like to pause for another couple of minutes so we can answer any questions that the participants may have for this part of the presentation. And actually, we have one right now. Will the datasets and test platform remain online beyond the end of the challenge to allow testing of future algorithms?
Yes, these datasets, together with the pipelines that will be made public, will be available on the platform even after the end of the challenge. However, the only thing to note is the subsequent analysis at the end of the challenge will not be funded by the challenge. That means that the users would have to pull them into their own workspace to run their own analysis.
Right, and I would also like to add that the evaluation apps, so the apps that run the underlying algorithms that do the comparison between the reference datasets and the submissions, are public applications, so people are still going to be able to use them on the data. So another question we have, Ken, is can I use the data for my benchmarking and publish my results of the benchmarking? So I think they mean, can they download the data, use them for their own research and publish the results?
Yes, as we mentioned, the data set is freely open to any participant, and we would like to, we would definitely encourage them to contact us so that we can work together. But at the same time, we are not restricting anyone from downloading the data set and using it as long as it is properly attributed.
Okay. So another question is, how did you ensure that there is no contamination?
So based on our work with Jeremiah Faith, he has taken a lot of care in terms of ensuring that they only work with one donor's sample during the week itself, and they have the right stringent protocols in ensuring that there is a minimal risk of contamination.
Okay. So are the strains used in the challenge and the testing ground the same?
No, the strains for the training data set and the challenge data set are completely different. They're not overlapping in any way.
Okay. All right, so I think that's all in terms of questions. So now we can move to the third part of the presentation, which is a live demo, which will be given by Sam Westreich, who is a microbiome scientist for DNA Nexus. So Sam, if you can, so Sam can start right now, actually, yeah.
Yeah, thank you, everyone. Thank you for joining us this morning and listening in. So I am going to walk through how to join this challenge, where to find the initial data that's provided for the challenge, how to submit preliminary results to the testing ground to see your performance, view the performance of those submissions, and then finally how to submit the final results to evaluation. So we will be moving through this challenge overview, starting with joining the challenge, looking at where the training data sets are and how you submit an analysis of those, and then comparing with the community, and then finally moving on to show how to analyze the challenge data set. So now I will share my screen. And we should be able to see here that I am logged into Mosaic with an account, and I'm just on the main page. So here at the top, I can find my workspaces, access to tools and applications, both those that are public and those that I built, discussions tab, I can form groups, and I can finally join challenges. Going over to this workspaces tab very briefly, I want to iterate that these are each personal virtual environments. So here it's possible to experiment with your data, to run analyses on any data inside these workspaces, and this is all private unless it is shared with other users. Now, if I go back to the main page, as has been mentioned earlier, strains number 2, this clinical strains detection, is not the only challenge that's running. We also have strains number 1, which will be closing very shortly. And if anyone is interested in another upcoming challenge and standards challenge, you can learn more here. But we are going to go to the strains number 2 challenge, which brings us to this overview page. In the overview and in the instructions here, we're able to find everything that's been covered in this webinar. So if anybody forgot anything from here or wants to go back and review, all of this is once again provided here, including an overview, description of the data sets, description of the different methods for data analysis, how to submit results and the format for those result submissions, access to the evaluation app, and additionally explanation of all the different metrics that are used for evaluation. So if I come back up to the top, I can click Join Strains 2. I agree with the rules as I've heard, and now I've successfully joined. And so once again, in case anybody forgets where their data sets are, there are links here where I can click this link and it brings me back over to the workspaces where I can find all of the data, in this case for the training assets. There is another workspace which will contain your data for challenge assets. And so I can run anything on these files. I can generate a submission. And when I am ready to see how well my submission scores, I can come back over, I can select the strains number 2 challenge, and over here on the left, I can click on Testing Ground. Here on Testing Ground, we can see that I don't have any submissions. And so let's say that I've built my workflow, I have run it on the training data, and I've created a submission, and I want to know how well it does. I can click here, and as explained, I can either choose to submit my data under my real name or any name that I provide here. So if I'm testing a pipeline, I might want to put the pipeline name, or I can choose to make the submission anonymous and keep the name secret. I need to provide a label. So a label for this might be test data set generated by pipeline number 1. I will select my results. So if I go simply to a demo workspace, I can select a results file prepared for this. And then here in the description, this is all done in Markdown format, and it's highly encouraged that you explain how you were able to generate these results. So this is a test data set, and then I can say these results and I might give additional details about the pipeline used to generate these results. Then, when I click submit, I agree that I am submitting this file, and this will bring me back to the testing ground. And so here, when I go down to submissions, when other individuals competing in this challenge submit their testing ground submissions, they will show up here for comparison. And if I click over, I can view my current submission, which is currently being checked by the evaluator application. Now I am going to jump over to staging, where here I can show what this will look like after a few other individuals have submitted. So here on staging, several different individuals have submitted their results to the testing ground, and I'm able to click on any of these submissions, and I can see the scores for that submission, including the adjusted RAND index, which is used for the final score. But I can also see how many true positives, true negatives, false positives, and false negatives I have, as well as these other metrics. And finally, I can see the ranking for each individual submission to determine how well it's doing in this challenge. Now, if you chose to submit your results with fractional numbers, if you chose to provide float estimates of confidence, then there will also be a precision recall curve generated for your submission. If you choose to provide binary results, where you only state with ones or zeros whether a strain is present or absent in a sample, this chart will not be generated. And you can also view your submitted files and re-download those, and you can view any description that's provided. Additionally, in this testing ground, if I go back to the testing ground, I can click up here to explore and see how well my submission compares to others within this testing ground environment. So here, once again, I see all the submissions from all users who have submitted to the testing ground so far, and I can see how their scores break down, the differences in true positives, true negatives, and false positives and negatives, as well as the breakdown in other metrics. I can also view the precision and recall charts for all of these different users overlapped, and I can toggle these on or off to view certain subsets. And finally, we have a leapfrog chart. So as different users submit to the testing ground, we will see which users are in the lead over time. So all of this is just for testing, but now let's say that I have finished with my testing data sets. I am very confident in my pipeline, and I am ready to make my challenge submission. Once again, over here on the left, we simply click on challenge submissions, and this format is almost identical to the testing ground. I click on submit results for evaluation. I can provide my name, or I can choose to submit my final results anonymously. I can provide a label. So this might be final submission. And then I can select a results file, add a description, and I will click submit. The one important difference to note here is that when I submit to the challenge, itself, these results will not be visible immediately. You will not be able to see how well you scored on the challenge submission, and you will not be able to compare your ranking to that of other users until after the challenge has fully ended. Now, after all of this, if you have any questions that come up, any issues, remember that you can always go back and you can find everything that we've covered today here in the instructions, including description of the data sets, access to the data, links to download the raw data if you wish to perform this on your own computer, description of how to submit results, a description of the format for the results, access to the evaluator app. So if you wish to download the evaluator app and perform your own evaluations of your locally analyzed results, you are free to do so. Description of the metrics. And then finally, at the bottom, there is a link where you can contact us. So if you have any other problems that come up, any issues, or you have a question about handling a certain part of the challenge, please feel free to use this form to contact us and we will respond promptly. And with that, I will pause, and if there are any questions, you can ask them now.
Thank you, Sam. So there are a couple of questions. One, how long does the evaluation take?
The evaluation app will take between 5 and 15 minutes. If I refresh this now, we can see that this test data set, which I submitted just now during the challenge, has already finished and has been approved. And so I can click here and I can view the results.
Great. So the other question is, how do we run the evaluator app?
So the evaluator app is a Python script which takes several files as input and provides the stores in an output file. When you download the app, you will get this script, and the script contains explanations of how to use it within comments, and it should also tell you. You can also find the evaluator app located under Tools as a public tool. Here it is right here. And so if you wish, you can run this app right on Mosaic. You can upload your result to Mosaic in your private workspace, run the evaluator app, and seed the stores for your private result without having to fully submit it and make it public in the testing ground.
So actually, Sam, can you go ahead and click on Run App so participants can see how that looks?
Sure. So when I click on Run App, here I choose my workspace, and I can choose either a demo workspace or one of the challenge ones. I would select my file that I'm going to use as my submission. So here is my example submission file. And then the answer key is linked here by default. So you don't need to fill that in. That will be provided as the suggested default. And then I simply click Run App. Once I've done this, I can now see for this application, there is a job running. If I select this job, we can see that this is currently waiting in the queue to be run. After a few minutes, this will change to running. And then once it has finished, the files that it provides as outputs will be here, which will include the stores that you would receive if you submitted this to the testing ground.
Great. Another question is, how can we get the underlying evaluation algorithm? So I think they mean the Python script that the app runs.
So if you want to see the underlying evaluation algorithm, if you want to see what that Python script looks like, that can either be accessed by downloading the application here in the challenge and viewing it that way, or we will have this on our public-facing GitHub. So this is the DNA Nexus GitHub under the folder for Mosaic. If you look through there, you will find the script that is used for here publicly available, so you can view it there.
Great. So I don't think there are any more questions. So we'll go back to the presentation. So Keng can talk to us about the upcoming standards challenge.
Right. Can you put the slides back? Yep, thank you. So now I just want to take
Keng, let me give you remote control first.
Right. Okay.
Go ahead.
Now I just want to take a few minutes to share on the standards challenge that we will be launching in a few weeks' time. Trying to move this. Okay, thanks. So besides the two strange challenges that we have launched that are purely computational challenge, we are also focusing on the standards, the issue of standards and protocols. And so we'll be launching this community effort called Standards. There was several key studies that have looked at the impact of protocols such as sample collection, storage, DNA extraction, and how they have affected the taxonomic readouts of NGS-based microbiome profiling studies. And in some cases, the variability that arises from the key differences in the protocol can be even larger than the biological variability. And this impacts not only the reproducibility of the results, but also more importantly, they can result in very different conclusions And there were two key papers that were published last year by the MBQC, the Microbiome Baseline Quality Control Consortium, as well as a very similar other consortium in Europe. However, both are limited in terms of their size of the study and also in terms of the breadth of the protocols tested. Hence, we have embarked on this community effort to capture the diversity of protocols out there. And how we are planning to do so is in the following manner. So we have created a set of 7 samples comprising 5 homogenized stool samples prepared by our partners, the BioCollective and DNA Genotech, as well as 2 DNA mixture samples that is contributed by the NIST team led by Scott Jackson. And we have 700 sets of these samples that we will be shipping to any lab in the world for free that are willing to participate in this community effort. And participating labs will then process these samples using their own protocols and sequence them. Then they can upload the sequencing data as well as the protocol metadata to Mosaic, which will then process their data and provide participants with 2 key pieces of information. Firstly, how does their protocol differ and compare against all the other participants? And second, in terms of a PICO-A plot of the results and how their results can be different based on different samples, as well as the different protocols between their samples and the other participants. And these samples will soon become available in a few weeks' time. And so I encourage anyone who is interested to sign up on the expression of interest page so that you can be notified immediately when the samples become available. And with this, I thank you all for your attention, and we'll be happy to take on any more questions out there.
Thank you, Keng. So there's one question: will the sequencing reads and metadata be available for download?
So yes, we are intending to make the sequencing reads and protocols available on the platform on a request or open basis. So we are still working out the terms of agreement for this challenge, but it is our fullest intention to make sure that any researcher can make full use of these the data, provided that they have been. I mean, we also understand that there are protocols that might be proprietary, so will be working with the community to figure out how we can best capture the diversity and at the same time will not compromise on these proprietary protocols.
Thank you, Keng. I'll just wait another 1 minute before we close. If there are any more questions, we can answer them. All right, here's one. Is there any limitation in which methods I use?
So are you referring, is the question referring to the protocols?
I am assuming it's referring to the sequencing for the standard challenge.
Right. So currently we are preparing that the analysis will be done using either 16S amplicon type analysis or shotgun. So if there are any participants that are going to try more other approaches like high-C or high-throughput single-cell kind of analysis, we'll be happy to work with them. But we think that these are going to be a small subset of the overall number of participants, and so they will probably require some customized form of analysis.
Another question: are there going to be winners in this challenge?
Currently, this is really designed more as a community effort than a challenge. So there will probably be no winner per se for this challenge that we can envision.
Okay. There's no more questions, so I would like to thank all the speakers, Dirk, Keng, and Sam, for taking time to talk with us today, and also everybody who participated in this webinar. So with that, thank you very much. Bye-bye.